Welcome to our second webinar in the University of the Sunshine Coast Research Unmasked seminar series. I'm Dr. Gemma Reid and I've been part of the organising committee for this series. I'd like to, on behalf of those participating in the session today, acknowledge the traditional custodians of the lands and waters upon which the university's campuses are located. I acknowledge their continuing connections to country and pay my respects to elders past, present and emerging. And I recognise that these lands have always been places of learning and teaching for Indigenous peoples. So tonight's webinar is all about vaccines and clinical trials. Our session facilitator is Professor Stefan Riek, and he'll be facilitating a discussion with our expert, Professor Scott Kitchener. So by way of introduction, Professor Stefan Riek joined USC in April this year as our Dean of Graduate Research after many years at the University of Queensland. His research is in brain plasticity and how the brain controls movement. One of Stefan's current research projects is a trial looking at how effective physical exercise can be on delaying age-related cognitive decline by promoting neurogenesis, or the growth of new nerve cells in the brain, even in the elderly. He has over 100 publications and has won more than $10 million in research grant income. Then we have Professor Scott Kitchener, who is a public health physician and principal investigator in the COVID-19 vaccine trial currently being conducted at USC's Clinical Trials Centre in Morayfield and Sippy Downs. Scott has extensive experience investigating vaccines to fight diseases including dengue fever and smallpox, and has spent time in the Australian Army gaining a specialisation in tropical medicine. He was stationed at Papua New Guinea and then Bougainville as part of the Army Malaria Institute, developing new treatments to combat malaria. He joined USC's Clinical Trials Centre in Morayfield earlier this year. So please enjoy the following discussion between Stefan and Scott.
What are some of the symptoms of COVID-19, particularly those that might be different from what we might normally expect? And do we know why the severity and recovery from COVID-19 can be so variable?
Well, I think there's a lot of things we don't know. A year ago, we wouldn't be talking about this virus. We would probably speak historically about how over a decade ago, we had a severe acute respiratory syndrome that arose from a coronavirus. And we could talk about the 4 common coronaviruses that cause a flu-like illness. But this particular virus we didn't know about, and I can't really tell you why it's so different. There's a lot of theories, and it's coming together. But very clearly, the symptoms are quite variable. We looked at the first 1,000 cases here in Queensland, and of those, the vast majority were actually managed out of hospital, and so that's a very encouraging thing. However, there were a tip of the iceberg, 1% or less of people who were very severely ill, and we've had 6 deaths in Queensland associated with the virus. It's got an enormous spectrum of severity and of symptoms, although primarily it is a respiratory illness, but people present with gastrointestinal symptoms. It's clearly got what's called a neurotropism, and that's probably best seen in the sense of smell and taste loss that occurs with the syndrome, and that's quite a resilient, common symptom. So the reason that you get a loss of smell and taste, particularly smell, is that at the very top of the nose, the brain is very close to the surface. There's a very thin layer of bone called the cribriform plate, and through that come nerves that give you the sense of smell. Clearly, the virus is getting into there and is upsetting those nerves. So that may well also be related to the longer-term symptoms that we're starting to see, which is very much a frontier of this clinical science. There appears to be what's being called COVID long, but it's essentially a longer-term subacute or even chronic illness associated with infection with the virus, and that seems to have fatigue syndromes associated with it, as well as longer-term respiratory symptoms.
And so I guess with the novelty of the virus, we still don't know how long some of these symptoms might continue and how long it will take some people to recover completely.
Yeah, so there was some data came out of the UK recently that considered that at least 10% of people will still have significant clinical symptoms 30 days after probable onset of clinical symptoms. Some people are still having symptoms at 3, 4, and 5 months. We don't really have many people who've had symptoms more than 10 months because it's only been around for 10 months.
That's right. So there's obviously, you know, an enormous amount of resources going into developing vaccines against the SARS-CoV-2 virus. Can you tell us a bit about some of the different types of vaccines that are in development?
A huge number. Over 300 vaccines are currently in development, which is incredibly encouraging. A number of the vaccines that are a long way along the development program are based on existing platforms. And so in vaccine research, commonly a standard platform is developed, that is to say, a way of doing the vaccine, and then the antigen, the signature of the particular virus that or bacteria that you're developing a vaccine for is inserted onto the platform in some way. That's really allowed a lot of vaccines to move through quickly. So those platforms can vary from adenoviruses, so a different virus is the Oxford vaccine approach, where they have that virus producing an antigen that's comparable to the spike protein, the spiky bits that appear on the coronavirus, so that essentially the virus that you get infected with, which isn't pathogenic or shouldn't be pathogenic, creates an immune structure, an immune response to a site on the coronavirus. So theoretically, after you get vaccinated with that vaccine, your immune system would recognize a coronavirus. More simple vaccines are the one that we're testing at the University of Sunshine Coast, which is essentially a recombinant spike protein vaccine. So this is the fewest moving parts, and I think when you're trying to move quickly and nimbly, then the fewest moving parts it's definitely the way to go. So this vaccine is essentially just the protein that appears like the spike protein on the coronavirus, and we put that in with an adjuvant. The adjuvant essentially stimulates the immune response, and the immune response that occurs would then subsequently recognise the coronavirus with that spike that is so typical of it. Between those two, there's everything. There's even oral vaccines, topical vaccines, vaccines that use different means of introducing the spike protein. Commonly, it's the spike protein because that's the most obvious part that you want your immune system to respond to. But in the context of 300 vaccines, and I've been in vaccine development for 20 odd years, and I've never seen such an array of vaccines going forward. So it's incredibly exciting, but it's also very encouraging in that out of those we will get a vaccine.
Yeah, I was going to ask you if it was usual to have so many groups trying to develop a vaccine, and why is it that it's just this particular virus that has gotten everybody excited in terms of developing a vaccine? Is it because it's been so virulent and so widespread and resulted in a pandemic?
Well, I think there's that, but it's the secondary effects of the pandemic. We have governments that are willing to invest. The Gates Foundation supports a lot of vaccine development, particularly in high-profile vaccines. So they've worked in HIV vaccines as well as a number of other ones. The model that they use is to provide some financial certainty because developing a vaccine can be extraordinarily expensive. It's not done unless there's a market at the other end. If you can give a certainty of that market at the other end, then you're more likely to actually get a vaccine be developed. And that's like anything. the consequence of a pandemic where governments are recognising that the presence of the virus is economically damaging as well as damaging to their populations is that they're willing to assure that market at the other end. And I think our government has been the same in that we've already pre-purchased vaccines associated with the Oxford group as well as the CSL University of Queensland vaccine. Now, not only does that mean that you have a lot of people interested in getting into that market, it also means that the market can perform differently. So previously, we have had vaccines where we would develop up a target. We would then look at a couple of candidates, a couple of ways of doing it. We would then move through 3 phases, which we can discuss later. And then after we're really sure that we're going to get a vaccine out the other end that can go to market, we'll then develop the manufacture, which means it's all linear. What's happening here is that we're seeing a vaccine development program that is in parallel. Now, you wouldn't do that normally because of the risk of the vaccine not actually going to licensure and not having a market. Obviously, you wouldn't invest in manufacture.
Yeah. Okay. Look, we've got our first question from the audience, and it's, do some people have a natural immunity to the disease?
Possibly. I don't think it's a natural immunity. I guess it could be called natural because it's exposure to the wild virus. That's about as natural as you get. It's not a great way to get immunity. It doesn't appear that there's a significant cross immunity. So some of the people listening will be aware that you can get immunity to one virus from another. And I guess the classic is that people who had been exposed to cowpox were originally recognised as potentially having some protection against smallpox. And that really is what led to the whole smallpox vaccination development 300 years ago. But we don't seem to see that the common 4 coronaviruses provide a significant amount of immunity to this one. So I can't really say that we're seeing a great deal of natural immunity. I think if you look at immunity in the broader sense of are some people less likely to become infected or become ill? Yes, for sure. We know that there are vulnerable populations, therefore there are less vulnerable populations. We know that some people get a very serious illness and some people don't. We don't know why completely.
Yeah. And just for everyone's benefit, can you tell us a bit about what the 4 common coronaviruses are?
Well, they basically are just 4 viruses that we have known to circulate, and they cause a minor influenza-like illness in humans. There are a number of other coronas that circulate, particularly in animals. Doesn't seem to be a great deal of cross contamination or transmission, although obviously this particular corona, SARS-CoV-2, has crossed over from animals.
And so once the hard work in the lab is done in terms of developing vaccines, what are the next steps that you have to do as a clinical trial to make sure that they're then safe and effective for the public?
Yeah, look, I think to cover 2 other coronas that exist that people may have heard of, and that is SARS and MERS. Those viruses occurred and caused smaller pandemics that were associated with public health emergencies, and they had vaccines developed, and those vaccines got through phase 1. So phase 1 is essentially where you have developed up a candidate vaccine, and that's commonly developed in an animal model. And then phase 1 refers to phase 1 in humans, once you've got all of your preclinical data. And that would be everything from whether or not this is safe in probably at least 2 animal models, what sort of immune response that you see in mammalian models, whether or not you can produce it and make it pure, whether it can be produced and administered and stored, for instance. So some vaccines are still at using -80, which clearly in the context of a population vaccination program is totally non-feasible. So there's a lot of work goes into the vaccine before you get to phase 1. And phase 1 is the first phase in humans, and typically it's a small number you would see, probably no more than a couple of dozen people administered the vaccine in a very controlled environment. Very often it's looking for some sort of a dose calculation. So you'll commonly see a small dose given to 1 or 2 people. They'll be watched very closely, and then there'll be larger doses given, or more people get that lower dose, and then a step up to a larger dose, and then a larger dose again. 3 doses is not uncommon, and that gives you some idea about how safe this vaccine is. Typically, in a phase 1 study, there'll also be some specific outcome variables. So, and obviously in this case, we're looking for the presence of antibodies stimulated by the vaccine. So that's the main game of phase 1. It's predominantly safety, some dose finding, and early efficacy data. And bear in mind that the presence of antibodies infers that you'll get protection from the vaccine. At this point, we're not actually giving the vaccine and then allowing people to be exposed to the virus. Now, that's a little different to the way you would develop a medication for blood pressure, for instance, where you may start with, you may even start with people that have mild high blood pressure, and then see whether or not it brings the blood pressure down. A better example is probably oncology drugs, where you would commonly give a medication in phase 1 to people that have the cancer, and you would be looking for some true efficacy, whereas in this case we're looking for a biomarker, the biomarker being neutralizing antibodies. Yes. Then we would move to phase 2, and phase 2 is where we're at in the University of Sunshine Coast trials, and this is where you give it to a larger number of people. Commonly, you have the dose that you intend on using. You're still very much looking for expanded safety data and tolerability. So we're very interested in how people respond, and we encourage them to tell us how it feels to have the vaccine. And then we're looking for expanded data on their response. And then phase 3, in inverted commas, simply it takes it into the field, where you're looking at significantly greater numbers. So while we're doing the trial, the phase 2 trial that University of Sunshine Coast is involved with is it's part of a trial of 1,500 people. And that's a reasonable number for phase 2. In fact, it's probably a large number for most phase 2s of vaccines. But the trials that are occurring for phase 3 vaccines, where we're actually looking to see whether this vaccine works in the field, are numbered in the tens of thousands. The size of those vaccine trials are really dependent upon statistical power. So if you can roll that vaccine out in a place where there is a lot of the virus and a lot of the disease, then it gives you the opportunity to give it to fewer people to actually see a true field effect. Nonetheless, you still want a large number of people because you want to expand your safety and tolerability data as well. But in terms of, does this work in the field? If we were to trial a phase 3 vaccine here in Queensland, we would be doing it for decades at our current incidence, which is why you'll find that many of the phase 3 trials are done in real hotspots, South Africa, New York, Brazil, those places.
Understand. A couple more questions from the audience. There's 2 that are kind of similar, so I'm going to try to summarize them. And they're asking about mutations of the virus. Are there mutations, and will a single vaccine protect people from the various mutations that might arise from the virus?
There are definitely mutations. I mean, probably the most tangible evidence of that is that in Queensland, we use a lot of shoe leather epidemiology, where we actually go out and we talk to people and make contacts and map outbreaks in that manner. In Victoria, they're using a lot of what's called genomic epidemiology. So they're actually doing a genomic analysis of each virus, and they're able to determine where that virus came from in terms of other cases that had a similar genomic appearance. So clearly, the virus has mutations. It really is about the rate of mutation. So, for instance, I worked for a time on hepatitis C vaccines, which are extraordinarily difficult to create because the virus in one individual will create tens of thousands of mutations immediately, virtually, in a very quick, short time. This virus does not seem to be doing that. An intermediate position would be what we see with influenza virus, where the influenza virus on a year-to-year basis drifts in its antigenic appearance, so the spots that appear for the immune system to respond to, which is why we need a new vaccine every year. And then every decade or so, it has a big shift, so a major mutation, which catches a lot of people by surprise, and obviously we need a new vaccine. At this stage, we don't think that this is associated with those massive shifts or the great difficulties of a Hep C vaccine, for instance. But again, it's too soon to tell. This has been around 10 months. The immune response is really quirky. There are some really difficult diseases in immune response. Tuberculosis is still a big black box to us in terms of the immune response. But this coronavirus seems to, we look at people who've previously had the virus, the wild virus, and their immune system seems to respond. It responds very variably, but it doesn't seem to last either. So we know that there has actually been people who have had second episodes, even though they're confirmed as having it first. Now that creates some serious challenges, considering that it's the presence of an immune response that we're depending upon for the vaccine to work. So I'm also a part of a research project that was recently funded that's looking at mapping the immune response. So we need to know so much more about it as we evolve the vaccines into second and third generation agents.
So what would a vaccine program look like once we say had arrived at a vaccine, or even multiple vaccines that passed through those trials and were shown to be effective?
Well, one thing that is going to be unusual for us is as a public health physician, and I became a specialist last century, so it's been a while. Rarely have I ever had the circumstances where you come to me and I say, well, we need to vaccinate you against this, and here's your choice of vaccines. That's very uncommon. And if we do have choices, it's one or two vaccines. So, I can, if we vaccinate you for tetanus, there's a couple of different ways that we can do it. We've never really had the circumstances where we will be able to tailor the nature of the vaccine. So that's going to be a very interesting aspect of the vaccine rollout program. And the tailoring will be, I think, largely to the major differences in our population. So I think what we'll have to do is we'll have to be looking for a vaccine that's best suited for older folks, the vaccine that's best suited for people with certain common morbidities. So, there may even be a particular vaccine for people that have chronic respiratory disease, and that may be a different vaccine to the one that we give to healthy 30-year-olds, for instance. There's highly likely to be a different vaccine for children, and that already exists in that we have different vaccines for children. So that's going to be an interesting aspect of it. The other aspect of a vaccine program that we'll have to roll out is how quickly we roll it out. There's 5.1 million Queenslanders. The vaccine that University of Sunshine Coast is involved in developing requires 2 shots. So now we're talking about 10.2 million people that are going to have to receive vaccines. That's not an insignificant task. And so there'll be a prioritisation matter as well. Who do you give it to first? Do you give it to people that are vulnerable? Do you give it to people that are exposed more? Do you give it to people that are more likely to be asymptomatic transmitters? All of these things are to be determined.
Yeah, still lots of questions. I wanted to ask, from a personal point of view, I'm looking forward to getting the vaccine because I don't want to get sick. But why is it important that this be widespread in the community, not only in Queensland and Australia, but really globally?
So there's a concept that you discuss with public health students and called herd immunity, which, up until I think this year was really a quirky thing that you only discussed in public health. And now you have Boris Johnson speaking, semi-sensibly about it. So that's one of the main reasons that that we look at getting to a point of enough people having some form of immunity that we can stop transmission. Now that varies for different viruses. For more difficult viruses, so polio and measles, we need a very high herd immunity. It's estimated that the herd immunity necessary to stop transmission of SARS-CoV-2 in a community is probably in excess of 70%. We still have to figure that out. I mean, that's based on modelling and projections and assumptions. So that's one reason why you would want to get to a fairly high level of vaccination, because it's better to have the population vaccinated to develop their immunity than to let them get the disease. And we can see in countries where there's been a very high transmission rate that you still don't get to an adequate herd immunity. I think in nations like Sweden, where there's been a conscious permission for the virus to be transmitting by having fewer countermeasures, or in countries where countermeasures haven't really been possible because of the community, you still don't see more than 5 or 10% of the population who are immune. So they've not achieved herd immunity. So clearly vaccination is the way to achieve herd immunity, and herd immunity is actually a concept that came out of vaccination. Yeah. And then to really put a caveat on that, a number of the vaccines that we're developing don't create sterile immunity. So these aren't vaccines necessarily to prevent you getting the virus. They may be vaccines that prevent you getting the disease. Now, the consequence of that is we may then have people who are still transmitting the virus, even though they're not manifesting the disease. Hugely problematic. And you see that in some existing diseases. So, for instance, the tuberculosis vaccine is not about preventing you completely getting the bacillus. It's about mitigating the disease that you get.
And do we have any indication yet on how long the vaccine would stay effective?
No. These are very difficult things. I think that's going to be. We know that the number of platforms have been used, are effective in delivering an antigen and engendering an immune response. But the nature of the immune response, the kinetics of the immune response, is commonly associated with the actual agent itself. What we do know about coronavirus is that people don't sustain their antibodies. They may well sustain the cellular immunity, the cellular response, and they may well retain the cellular memory. So, for instance, I worked on a JE vaccine. We're able to see people lose their antibodies and therefore need a booster. But when we gave them a very small amount of the vaccine, probably not enough to actually give them immunity, their response in antibodies was really significant, which means that they had a cellular memory. And it means that probably, if they were exposed to the wild virus, that cellular memory would respond as quick as that, and they would have antibodies and they would be protected. So these kinetics of humoral or antibody immunity and cellular memory and cellular immunity are yet to be worked out.
Okay. And so do you, another question from our audience, do you think that a vaccine will eventually be able to eradicate the virus, or is it something that we're really just going to have to live with in some way, form or other?
Look, I think the eradication question is a fairly simple question to answer, and that is that have we ever been successful in eradicating a virus? We attempted with incredible effort to eradicate smallpox, and yet there were perverse reasons to maintain smallpox. I think to think that we'll eradicate it is optimistic, and I think just unrealistic.
if we can eradicate it, hopefully we can get it under control. How long do you think it's likely to take for the production and distribution of a viable vaccine, so that we can get enough global immunity so that I can go back on my overseas holidays and travel again, and will there be these measures put in place to continue to monitor that?
I think that's the question to ask. When will life get back to normal? When will we be able to accommodate this virus in our normal lives? And will those normal lives then be comparable to what life was like last year? I think that we'll have a vaccine. We'll have a number of vaccines next year. I'm sure that's going to happen, given the number of candidates and the effort and the successes to date. When do we have a vaccine that the entire world will have? I think that's a long way off, because there are enormous inequities in health globally, and we're probably further down the path on this particular disease than we are many other diseases, because the vaccine programs have been founded on bodies like CEPI, which is a collaboration that was developing platforms. It was and University of Queensland, for instance, vaccine that UQ and CSL are developing was part of that. And part of being in that vaccine development program is that you will make the vaccines available to countries equitably. Compare that to what is a great concern of doing business with American vaccine companies, and particularly if they are specifically American, is whether or not there will be vaccine development that then provides that vaccine back to the nation that does the development. And this is a common issue in public health emergencies. The ethics of public health emergencies, where you get research that's done in the locations where the emergency is very acute. Commonly, the research is done with the investment of developed nations who are fearful of being exposed to whatever agent is causing the emergency in that core nation. But the ethical issue is, do they then provide all of that back to the nation where the risk is greatest? And I think we saw that with Ebola, that a lot of countries were very fearful of Ebola, developed a lot of research around it, including early stage vaccines, but it wasn't clear that those vaccines were going to be made available back to the countries, or for that matter, all of the knowledge of countermeasures and management of the disease is made available back to the countries where it's most needed. So that's going to be a big issue with vaccines. So I think a global vaccination program is still a fair way off, but I think we're further down the path to getting that. In between, when can you travel again? I think we can look at other vaccines where there's been vaccine passports. So, for instance, you may recall it's probably 20 years ago now that you couldn't go into parts of Africa without a cholera vaccination certificate, and that was really about ensuring that people were safe when they went in, and that cholera wasn't transmitted by people who were moving around. Another great example is the yellow fever vaccine. So Australia is receptive to yellow fever, but is yellow fever free. And that means to say that if yellow fever virus is brought in, it would circulate in Australia because the vector is here, which is a particular mosquito. So consequently, if you go to a yellow fever endemic area, a country that has yellow fever, Australia won't let you back in the 10 days after you've left there, because the possibility is that you have the virus, and therefore you have to have the vaccine before you go in and have that certified to come back directly into Australia. Obviously, one way around that is to go to a country where yellow fever can't circulate, spend your 10 days in London, for instance, then fly back to Australia. So while those two examples are, I think, gives some idea about informing what a COVID vaccine passport may look like. Obviously, you can't do the yellow fever shuffle by going out to a non-receptive area and then coming back into a receptive country not being vaccinated. But I think we will start to see people travel with certification of some sort. Now, the other problem of this is that with yellow fever, we know that it lasts at least 10 years, the vaccine. With cholera, you could be pretty certain it lasted a year. This one, we don't yet know. It's clearly more complex. And so that's going to be a difficult aspect of the possibility of a COVID passport.
Yeah, for sure. Another question from our audience, a bit more specific around the USC trial you're involved in. Are you able to talk a bit about the demographics of the participants in that, and are you doing a wide range of participants or specific age groups or anything like that?
Yeah, look, that's a really good question, a very sensible question, in that many drug trials will focus on a very narrow age range of people who aren't sick, because obviously people that are older or people that are really young, you get into all sorts of troubles of them having health issues, and that being attributed to the drug that you're developing. And this is a major ethical issue in clinical trials globally. It does occur in vaccines in that you typically won't try to vaccinate people who are older because their immune system is starting to become less effective. And so you don't necessarily want to have your vaccine looking less effective because you're immunising people who are less able to mount an immune response. Pleasingly, the University of Sunshine Coast clinical trials for the COVID vaccine that we're working on have had people engaged up to the age of 85, which is really impressive. I mean, that meant that we've had to work very hard to ensure that we're giving people a vaccine safely. Obviously, we can't just have any 85-year-old come in, because by 85 you've tended to accrue a few health issues. And we wouldn't want to be careful not to be making people sick with what we're doing. But the fact that we're able to start to look at people who are in that known more vulnerable group of over 65, I think is a terrific direction.
Yeah, that is terrific. Just wondering, just a bit more on your sort of background. How did you end up getting into developing viruses, clinical trials for viruses? Tell us a bit about how you landed into this area.
I guess the best advice I'd give to students is get a great education. You're doing that now if you're at USC. And then when doors open, go through them. When opportunity knocks, answer. So I was working, I'd done a little bit of work on vaccines as a medical practitioner. And I was very interested in public health medicine. So I specialised in that area. But I was in the Army, and I was in the Army when we went to Bougainville and Timor. We got hit very hard by malaria. So I got involved in the Malaria Institute. We were developing anti-malarial drugs, and we developed 2 new drugs, both of which are on the market now, Malaria and tefenoquine, during the Bougainville and Timor operations. And that was really quite unusual to be able to do clinical trials in Timor. Was a frank war zone. That brought us to the attention of other pharmaceutical companies, and particularly in those operations, especially Timor, we got hit badly with dengue. So consequently, we looked around to see whether we could get a dengue vaccine. We'd also had issues with, in the mid-90s, Japanese encephalitis coming down from Asia. And in 1995, the first case occurred in Torres Strait. In 1998, the first case was identified on continental Australia. So we also became involved in developing a new way to do that vaccine. And we were in big trouble with the JE vaccine because it was a rough old vaccine. It was a vaccine that was developed in the brains of mice in Japan. And obviously, eventually, the Japanese company that was doing this decided that they were getting out of this business because it was all too hard. And so Australia had a set stockpile. And then we decided to deploy into JE endemic areas like Timor. So suddenly we had this set stockpile, and we went, well, we're going to have to do a lot of vaccines, and we're going to deplete the stockpile, and we still have to protect the people of Torres Strait. So the first thing we did was develop a new way to give the vaccine with a fifth of the dose. So that was really my first vaccine trials. It was out of necessity. We were sitting around having a few beers, as it turned out, and can I tell you, some of the best research ideas come when you're talking to other people. Research never should occur in isolation. It's with people. Ideas get bounced and ideas come up. And we got this great idea to do intradermal vaccination of the existing vaccine. That worked very well. We sent entire battalions to Timor protected against JE using a fifth of the vaccine stockpile. We had success with that, and then when we got hit with dengue, we went, well, look, we've got some experience in this space. We knocked on the door of a number of companies, and Aventis Pasteur came to the party and helped. We partnered up to start to develop dengue vaccines, and those vaccines have now moved to licensure in parts of the world. But then I finished my doctorate on JE vaccines and did a postdoc in Cambridge, working on the smallpox vaccine stockpile. Some of the listeners may actually be too young to remember. There was a number of aircraft that went into a couple of buildings in the United States, and at that time, one of the great concerns was that there was weaponized smallpox around, and so the United States wanted to get a stockpile of smallpox vaccines. I was very fortunate to get in with a research group that said, yeah, we can get you 200 million doses of that vaccine, and so we did. And that gave us a stockpile of money, and it was a merry bunch of people who then thought, well, we've got this money, let's do some good and let's develop some vaccines for the developing world. So I could never have planned that when I finished my university course, but opportunity knocked, and I went through, and it's probably the best advice I give to any students is that when the opportunity arises, Alexander Yersin used to say, actually his mentor, Louis Pasteur, used to say that fortune favours the prepared mind. So study hard at uni, prepare yourself, and then keep your head up and watch out for those opportunities, and a path that you would not expect to arise will find your feet.
Yeah, I think that's really good advice, Scott. I think, being open to opportunities and recognizing them is pretty important. And I think, we all have similar elements of stories that have determined our path, often unpredicted. Speaking of predictability, do you think that a pandemic like COVID-19 could have been predicted?
That's a great question. I think a pandemic of a respiratory illness was predicted. We certainly were planning on, and have been planning on, an influenza pandemic for probably decades now. We definitely had a H1N1 influenza epidemic a few years ago. And I think one of the ironies is that we were expecting a respiratory epidemic. I think we coned down on that was going to be influenza, which probably didn't serve us well in that it wasn't transposable to corona. There were certainly people who have started to appear who were doing research on corona and were aware of it. And there's even some interesting exercises that were conducted of the possibility of a corona becoming epidemic. But I think all of the planning around influenza, which has a vaccine, which has a vaccine infrastructure, so the ability to quickly shift the vaccine to the new target, having a manufacture system up and running, that wasn't there for corona. So to a degree, we did ourselves a little bit of a disservice in planning for an influenza pandemic. And with respect to corona, we were certainly given warning. I think SARS-1, 2003, 2004, clearly that was a herald, and then in 2012 we had MERS, another corona. So yeah, I mean, it's very clever, easy to be clever with hindsight, isn't it?
It is indeed. And do we, I mean, have we got to the bottom of how this particular virus started in humans?
Look, will we ever get to the bottom? I think what we know is that it jumped from an animal. I don't think it's clear as to what animal that was. I think that that's often a very difficult question. You know, there's been decades of research on where viruses came from in many other diseases. So, yeah, I'm not sure we'll ever know. There's a lot of work gone into it, and I think it suffers from limited returns to a degree.
Yeah, I understand. I think last question from me, and that's really just, I'm interested to find out a bit more about your work on malaria and trying to combat malaria, although not a virus, but if you could put as many resources that have been going on the last sort of 10 months fighting COVID to fighting malaria, what do you think you could do to?
Well, that is a good question. I mean, another great question along those lines is the pandemic we haven't spoken about, which is the HIV pandemic. When you work in malaria, you get very jealous of the HIV people because they certainly, when I was working globally, it was a matter of the money that was being poured into HIV vaccines, particularly, which is a very difficult nut to crack, was so much more than malaria. And but then you meet the tuberculosis people. The tuberculosis people wish that they had the resources of the malaria people. All of these are wicked diseases. And look, I think we probably have the tools to do what we need to do with malaria, to be honest. The last drug that we developed, the Australian Army is a global leader in management of malaria and development of the latest drugs. And one of the drugs that we developed, tefenoquine, has an amazing half-life. It's got a 17-day half-life. It's got efficacy against the full range of the life cycle of the parasite. We could roll that out on an island, rid the island of malaria, including the reservoir, because bear in mind it's a bit of an easier nut to crack in that it's only a human disease. And then as long as we are ensuring that no new malaria comes in, so we're screening people and doing the right quarantine, we've ridded that island of malaria, and that's feasible now. So it's whether we choose to do it and how we choose to do it, whether the resources are allocated accordingly, or whether there's actually, is it better to spend those resources on something else? You know, there are bigger issues than malaria. Malaria is huge. There's 300 million cases a year and probably 3 million deaths a year. But that is dwarfed next to infant mortality. You know, there are nations where malaria is a big problem, where the infant mortality is 10%. Infantile diarrhea kills far more people. And that probably is a cheaper nut to crack. Should we be putting those resources there? So it's not an easy question, and perhaps discussing malaria at more length. And the University of Sunshine Coast Clinical Trials Unit is about to do a study on tefenoquine, so perhaps that's a topic for another evening.
That's right. Scott, I want to thank you very much. It's been a really enlightening and fascinating discussion. I think USC, Queensland, and Australia are really fortunate to have you working in this area, and our fingers are crossed that we can see a vaccine out in the public as soon as possible.
Well, can I say that it's a huge team effort. I'm just the silly bugger at the front. Guys like Luke Satuka and Sharon Rankin, the team at the Clinical Trial Centre are just, I've worked around the world in vaccine development, and the team that has been assembled at the University of Sunshine Coast is world class. It was extraordinary to have the opportunity to partner with them.
Thanks very much. And they are a fantastic team, and all the success to to everyone involved.
Thank you.
Thanks very much, Scott. I'll just thank you everyone for attending, and look forward to seeing you at next week's seminar, same time.